I met Shawn right as she was finishing up her undergrad. She wasn't quite sure what she wanted to do, but she came, or she thought she wanted to go to grad school, but she came and worked in our lab for a year, helping with the stranding program and a variety of other projects. After helping with that project and working in our lab for a few months, she contacted me about going to grad school and if I was interested in having her as a student. So like any good advisor, I did some internet stalking to see if she had the required skills to be successful in the lab. And some of the skills that are quite helpful when you're working with seals are to be athletic and agile. So I did find some evidence of this. Apparently in her high school years, she was a track star. So here you can see her doing the long jump. It can also be very helpful to be stealth. And she had a previous career as a spy. So that was a good sign. And it's also very helpful to have some water skills. This one I got a little bit more concerned about, but then after doing a little bit more hunting, it did look like she was confident in the water. So she had some of the basic skills that we think are important. Another one that's important in the lab is that you have to actually like the animals. Ideally, the animals you're working with, you're going to have some long days of working with the animals, processing the data. So you have to kind of have this love. And basically, there was an abundance of evidence of how much she loves animals. So these are kind of the less common house animals, but she also loves any dogs. I'm sure many of you guys know about her little rabbit bunny that got her through grad school. And if you've heard the rumor that if you spend too much time with your pets during COVID, it looked like she started to look like her pet. And then a little aside, and also just a thank you, I adopted a very scared dog during COVID. And Shawn was like the one person my dog bonded with. So when Shawn was watching my dog, she took this photo, you can see lying in the background there, as evidence that he was no longer hiding in the stairwell and was sitting next to her. There was also some evidence that she had good taste in role models. That's always a good sign. And then the final sign was that she'd already made a jersey with my name on it. So how could I say no? So I accepted her into the graduate program and she became a very valued team member. She initially thought she was going to work with harbor seals, but we recruited her to the dark side and she joined our NSF-funded elephant seal project. And she'll talk to you more about that today. She helped with all aspects of this project. She helped pack a ridiculous amount of supplies and inventory it. They tested out equipment and did some unsafe practices as they were transporting cages back and forth after cleaning. She also developed a lot of animal skills. Here you can see she's very good at holding down the drugged elephant seal. But in all seriousness, she did develop a lot of useful skills. She collected the blood samples. So when we were busy doing the anesthesia and the surgery procedures, she would collect the blood whenever it was needed. And then her and Daphne and Lauren all were essential in getting all of the lab work done. So great team. And this is kind of a picture of that elephant seal team before COVID when we weren't wearing masks. Because of her skills, she was often asked to help out with other projects. So she's helped out with weaner weighing and adult female procedures. She also was played an important role in our stranding program. So she was the stranding coordinator for a couple years. Here you can see as they are driving to the dump with a truck full of dead carcasses after our freezer broke, she still has a smile on her face. She was a great community member. She participated on our lab bowling team, always up for a lab barbecue. So just wonderful to have in the lab, hard worker, great colleague. And kind of to wrap up, she received 5 awards to support her graduate student research. She presented her results at 2 different conferences, Experimental Biology and the Marine Mammal Conference. And as a student here, she got 2 publications out as a co-author as work she started as an undergrad. And one of them was actually a paper with me. We started working together before we'd actually ever met. And then this paper should be submitted soon. So with that, I would like to say we are looking forward to hearing your talk. And you can take over.
All right. Thank you, Gita. That was an amazing introduction and very entertaining. So thank you so much. Let me go ahead and get my presentation pulled up here real quick. All righty. So again, thank you, Gita, so much for that incredible introduction. And thank you all for being here for the virtual defense of my thesis work, examining the plasticity of the dive response in relation to dive behavior of northern elephant seals, Mirounga angustirostris. All right. And with that, let's dive in. So marine mammals face this extraordinary challenge, right, where they have to actively search for and capture prey, but they do all of this under a breath hold dive. And marine mammals have very large oxygen storage capacity to handle this. So they must conserve these oxygen stores during their dives to maximize the time they can spend foraging. It's because these oxygen stores are key to maintaining their organ function and their energy metabolism during the dive. And specifically, managing their oxygen stores during the dive helps them dive aerobically, which ultimately allows them to maximize that time foraging. So what is diving aerobically? Well, aerobic diving means using aerobic metabolism during the dive. And simply put, aerobic metabolism is where the body uses oxygen to create energy. And over the course of the dive, they'll use this oxygen storage to create energy for their cells, organs, and muscles. But what if they begin to run out of oxygen in certain areas during the dive? Well, then the body resorts to anaerobic metabolism. And simply put, anaerobic metabolism is where, in the absence of oxygen, the body still does create energy, but this process results in the buildup of lactate, which is a negative side effect of anaerobic metabolism. And lactate, you might know as lactic acid. It can result in things such as muscle acidosis, and ultimately it can require these animals to have to recover longer at the surface, which minimizes the time they can spend foraging. And it also can make them vulnerable because being at the surface is where a lot of the predation on them can happen. So maximizing the time they spend using aerobic metabolism is the best case scenario for these animals. And there's this term keyed as the aerobic dive limit, or the ADL. And the aerobic dive limit is the maximum time these animals can dive before having to resort to anaerobic metabolism. So before they have a buildup of lactate in their blood above their resting level. And this dive limit has been measured in 4 marine animals, 1 penguin, and 3 marine mammals. And you'll see in this graph here from Hoyman and Ponganis in 1998, we have dive duration here on the x-axis and the post-dive blood lactate level on the y. And each of these animals has a kind of resting or regular lactate level in their blood for each of these different species. That's kind of the normal for them. But you'll see as dive durations begin to increase, each of them has this spike in lactate. And it's at this point where the dive duration exceeds a certain duration in time that they're no longer using aerobic metabolism, and they've switched to anaerobic metabolism and the buildup of that lactate above resting level. And it's at that point that is their aerobic dive limit. This has only been measured in 4 animals, like I said, but it can be calculated in other species. And you do that using the known oxygen storage capacity of that species, as well as their metabolic rate, to get an estimation of what that dive limit would be. And that's considered the calculated aerobic dive limit. So how do marine mammals maximize the time they spend using aerobic metabolism during the dive? Well, there's a suite of physiological adaptations known as the dive response. And what that is apnea or breath holds, peripheral vasoconstriction or shunting the blood to more vital areas of the body, such as the heart, brain, lungs, adrenal glands, and then also bradycardia or a low heart rate. And what these suite of physiological adaptations do is they lower that rate of oxygen consumption. They maintain physiological homeostasis, And overall, they prolong the duration of aerobic metabolism. There are some things that influence the dive response in these animals, and a key one is both the dive depth and duration of the dive, where it's been shown that longer, deeper dives result in animals having a stronger dive response. Stronger meaning there's more vasoconstriction, there's a lower heart rate, and we've seen this across decades in many species. Here in a figure from Hill et al. In 1987, you'll see on the x-axis here is the dive duration. On this lower y-axis is depth, and on the upper y-axis is heart rate. I've highlighted the 4 different dives in this figure here, with depth increasing vertically. And you see at the onset of a dive, the heart rate does drop significantly from the resting surface level. And it stays fairly low throughout the dive, increasing as the seal or whale or dolphin makes its way back to the surface. In this case, I should mention they were studying adult Weddell seals. So in this case, it was a Weddell seal. And that's consistent across the board with each of these dives. But you can see this last dive here is much longer in duration and much deeper than the other 3. And what you'll notice is that, again, with the onset of the dive, the heart rate drops significantly, but it is significantly lower than the previous 3 dives. And what they found is, yes, the heart rate is significantly lower in longer, deeper dives with Weddell seals. We've seen that with California sea lions and McDonald and Ponganis from 2014. They have this graph here showing dive duration again on the x-axis. That black line is heart rate, which is shown here on the left-hand side, and the blue line is depth, which is on the right-hand side. This top graph is a shorter, shallower dive, and this bottom graph is a longer, deeper dive. And what you'll notice again is that for the shallow dive, the heart rate drops to about 20 beats per minute, whereas in the steeper dive, the heart rate goes down to as low as 10 beats per minute or maybe even lower. So what they found again, yes, for the California sea lion, the heart rate drops with increasing dive duration. And lastly, just to drive this point home, even more recently in the largest species in the world, the blue whale, in Goldbogen et al., they found again that the blue whale's heart rate does drop with increasing dive duration. This is only one dive here, but you do see at the onset of the dive, the heart rate drops significantly and stays low throughout the dive, increasing as the whale makes its way back to the surface. So it's been proven over and over again that depth and duration do have a strong influence on the dive response. More recently, it's been shown that there's a degree of exercise modulation on the dive response. And what I mean by that is during bursts of activity throughout the dive, there will be a temporary weakened dive response. So instead of the heart rate dropping, it will actually increase, and there will be some vasodilation, so more blood flow. And if we think about the terrestrial exercise response, so picture a really intimidating predator going after its prey. Oh, maybe something more along the lines of this. Or honestly, just picture yourself exercising. What happens? Well, you have an increased heart rate. You have vasodilation, whether you realize it or not, your blood is flowing more throughout your body, and you begin to pant. So then how are marine mammals managing this, where they're exercising, but they're at depth and they have this strong dive response? Well, exercise modulation's kind of been a key component of this, where lately we've noticed that an increase or a burst of activity increases the heart rate temporarily, which leads to vasodilation or increased blood flow, which can send more oxygenated blood to areas such as the muscle that are working hard. And that enables that muscle to create energy using oxygen. So limiting that lactate buildup and creating energy without having to use anaerobic metabolism. We've seen this in a few species from Davis and Williams in 2012. They studied this in both bottlenose dolphins and Weddell seals. You have their stroking frequency here on the x-axis and their heart rate on the y. And you see with both of these species, as they begin to stroke more frequently, they have an increase in their heart rate. And they found that, yes, that with increased inactivity, the heart rate does increase. We've seen it in California sea lions with McDonald et al. In 2020, where they looked at this on a fine scale with California sea lions. Their activity metric here on the x-axis is the MSA of the sea lions. And on the y-axis, again, is that heart rate. It's color coordinated here by the depth of the dive. And you can see that with all of the different depths, there is a positive slope. So there is a positive relationship where an increase in activity resulted in an increase in heart rate. Interestingly, though, in McDonald et al., they found that this relationship between activity and heart rate was influenced by depth and duration, where an increase in the depth resulted in a decrease or weakening of that relationship between activity and heart rate. And you can really see it here with this, the deepest of depths that they measured, greater than 300 meters, that slope isn't nearly as steep as the others. And they ultimately realized that these sea lions still are prioritizing their blood oxygen conservation over perfusing the muscle area or delivering more blood that's oxygenated to the muscles. So they're still prioritizing conserving that oxygen in these longer, deeper dives. The last influence on the dive response is cognitive control. And there's a few examples of this, one being anticipation, where, for example, California sea lions anticipated longer dives by increasing their pre-dive heart rate, seemingly to onload more oxygen into their blood in preparation of that longer dive. And more recently, it's been shown in harbor seals that they actually have some control over their vasoconstriction, where they will redistribute the oxygen in their body in anticipation of a dive and during their dives. So ultimately, cognitive regulation of the dive response allows these animals to manage their blood oxygen stores based on anticipatory dive behavior and during their dive. So this led me to ask, what are the relationships among these variables? And to what degree do they influence the dive response at a fine scale? Fine scale meaning breaking up the dive into smaller increments to see what's going on throughout the dive. To answer this, I studied northern elephant seals, which are considered a model species in that they have been extensively studied, proven to handle instrumentation well as usually returning back to the same beach in translocation experiments. And a translocation experiment is where you capture an animal in one area, release it in another, and then hopefully that animal makes its way back to that original capture site. And that just ultimately increases tag retrieval for us. Northern elephant seals are also one of the deepest diving pinnipeds. On average, they dive around 350 to 650 meters for about 30 minutes, but they're capable of diving for over an hour to depths as low as 1,500 meters. So just really incredible divers, and they do this continuously. So they spend about 88% of their time at sea submerged, and they spend usually less than 3 minutes recovering between dives at the surface. So they're just really incredible divers. And I just wanted to mention, they do have a calculated aerobic dive limit, like I mentioned earlier, that's an estimation. And for them, it's been approximately estimated to be approximately about 30 minutes for adult females based on their oxygen storage capacity, as well as their metabolic rate. And they tend to exceed this calculated limit and still don't have to recover a lot at the surface, usually still spending less than 3 minutes recovering. So they're seemingly not building up a lot of lactate in their blood, though they're diving beyond this calculated limit. So incredible divers. And what do we know? Well, we know that their dive response is linked to their depth and duration. Again, they have a strong dive response when they have longer, deeper dives. They have an anticipatory tachycardia, where just before they surface at the end of their dive, they'll increase their heart rate. So by the time that they reach the surface, they're ready to begin that gas exchange and be super efficient on their recovery. And then they also, again, have that minimal recovery time between dives, usually less than 3 minutes. So what do we not know? Well, how does the dive response relate to activity on a fine scale? Again, breaking up that dive into smaller increments to see what's going on throughout the dive. And then how do all of these influences interact to affect the dive response? And lastly, are there any other cognitive influences on their dive response? And so in this study, I investigated the relationship between dive depth, duration, and activity on the dive heart rate of juvenile northern elephant seals, to determine the plasticity of their dive response. And my first hypothesis was that the dive heart rate would be lower with increased dive depth and duration, but it would increase with increased activity. Similarly, but at a fine scale, where I break the dive up into the 3 different phases and then break them up further into 20-second intervals, I hypothesized that at a fine scale, heart rate again would decrease with increased dive depth and duration, but it would increase with increased activity. And that this relationship between activity and heart rate would be influenced by the dive depth and duration, and that these relationships on a fine scale would be more pronounced than at the dive level. My third hypothesis was that the post-dive heart rate would increase with increased dive depth, duration, and activity. My fourth was that the surface recovery time, the time spent recovering between dives, would not change based on the SEAL's dive behavior. And lastly, that the pre-dive heart rate would increase with increased dive depth and duration. All right, so with that, let's get into how I studied this. Before I jump in, I just want to mention that this project is part of a larger National Science Foundation funded project that's titled the At-Sea Experimental Disturbances to Characterize Physiological Plasticity in Diving Northern Elephant Seals, with the principal investigators listed below. And that all the work you're seeing today was conducted under the National Marine Fisheries Service permits listed below, and that marine mammals are federally protected. So let's go into the capture and instrumentation part of my field work. We captured juvenile northern elephant seals at Año Nuevo State Park, which is about 45 minutes north of Santa Cruz, California. It took place in the years of 2018 and 2019 during the juveniles molting season, which was March and April. So we would go out to Año Nuevo State Park and look for a beautiful juvenile northern elephant seal. By beautiful, I mean like this little guy here, where they have not yet started molting. So northern elephant seals go through a catastrophic molt where they not only shed their fur, but also their skin. So this is a great example of a seal that would not be an ideal candidate for our experiment, though not ugly, just not ideal for us. You can see that we're losing some surface area to attach tags once they begin molting both their fur and their skin. So once we found an ideal candidate for our study, we would immobilize it with an injection intramuscularly of telazol, place it into a cage, and then transport it back to Long Marine Lab. And Long Marine Lab is located on the UC Santa Cruz campus, again, about 45 minutes south of Año Nuevo. Once there, the seals were allowed to recover from that light sedation that we gave on the beach for a few hours in their cage before we began our instrumentation procedure. And that instrumentation procedure was performed under isoflurane, 1 to 2% isoflurane gas and oxygen. And we attached the following tags: an Argo satellite tag, which tracked the seal's location out at sea, a very high frequency transmitter that we used to locate the seal once they returned back to the beach using telemetry, a daily diary behavior data logger, which measured the seal's 3-axis acceleration, as well as it was a time depth recorder, and then an Oxylog data logger, which was a state-of-the-art tag that had a few different electrodes attached to it. These electrodes were then inserted into the extradural vein of the seals. And the electrode that I used for my study was measuring the echocardiogram of the seals, or the ECG. And then an acoustic tag was attached to some of the seals, though I didn't use it in this study, so I won't be speaking to that tag, but all of these tags were attached using 5-minute quick set epoxy, as well as mesh and cable ties. Here's just a couple pictures of us attaching tags. You can see Daphne on the right-hand side using the 5-minute quick set epoxy to attach that tag to the seal. And then us fiddling around with those electrodes on the left-hand side, just trying to make sure that when the seal is released and begins diving, that those electrodes won't slip out of that extradural vein. Here's a picture of a seal after all of the instruments have been placed on him. And you'll see that large neoprene patch in the middle. That's just for extra protection of those electrodes again. And that patch at the very end of the seal would be removed before the seal was released. But They were able to recover in their cage after the instrumentation period for several hours after being under anesthesia. And then once they were ready to be released, they were released about 75 kilometers south of Long Marine Lab in Monterey, California, at either Hopkins Marine Station or a beach in Pacific Grove. They usually took about 3 to 5 days to make their way back to Año Nuevo, diving along the way. And once they were back at the beach, we recovered the tags by locating them using telemetry, sedating them again with a tilazole injection intramuscularly, and then removing the tags right there on the beach. The seals were then able to recover and then ultimately finish their catastrophic molt, shedding any leftover bits of glue or neoprene that may have been left from our instruments. So after the field season and we had all of our tags collected, I began the data processing and analyses. So just to orient you with my data sets that I used dive depth and duration to characterize the seal's dive behavior, and that came from that daily diary tag. I used heart rate as a proxy for the dive response, as it's highly correlated with the vasoconstriction part of the dive response. So it's a great way to measure the dive response as a whole, and that came from the Oxylog data logger. And lastly, ODBA, or overall dynamic body acceleration, was my acceleration metric where the activity and exercise of the seal was measured, and that came from the daily diary tag. So jumping into my dive processing, I used the dive move package in R to classify my dive start and stop times. I then characterized what I thought a dive should be classified as, which was a minimum duration of 20 seconds to a minimum depth of 5 meters. Anything that didn't meet that criteria was filtered out of my data. I then segmented the dive into its different parts, where the pre-dive was 30 seconds before a dive. The descent phase was the submersion to 80% of the maximum depth. The bottom phase was the time spent at greater than 80% maximum depth. And then the ascent phase was 80% maximum depth back to the surface. And then the post-dive was the 30 seconds after the dive. And I obtained the following dive behaviors: the mean and maximum dive depths, the dive durations, and the post-dive surface intervals, or that time recovering between dives. Jumping into my heart rate processing, I used that echocardiogram data from the electrode, which you can see here is a snippet of that. I have a heartbeat completely identified here from the P, Q, R, S, and T parts, but it was mainly those R peaks that I was using for my data. So I would identify a heartbeat by putting a green dot on the R peak in a custom written script in MATLAB. I would then use both the R peak as well as the R to R intervals for further processing. For those R peaks where I identified a heartbeat, I used those heartbeats to get my total beats per duration or my dive heart rate. So for the dive level, I had the total beats during the dive divided by that dive's duration to get the dive heart rate. Similarly, for the dive phase, I would count up how many beats were in that dive phase divided by that dive phase's duration, and so on with the pre-dive heart rate and the post-dive heart rate. I also calculated the instantaneous heart rate, which was 60 divided by that R to R peak interval to get beats per minute. I calculated the minimum and mean instantaneous heart rate for dives, as well as the mean instantaneous heart rate for my 20-second intervals, which was my fine scale analysis that I'll go into later. For my last data set, the accelerometry data, I actually calculated 2 different acceleration metrics: stroke rate and overall dynamic body acceleration, or ODBA. And as I mentioned, ODBA ended up being my acceleration metric, as it seemed to be the best choice, in that stroke rate was positively correlated to ODBA with this Pearson's correlation test. And ultimately, ODBA does incorporate the amplitude of the seal's acceleration, as well as I found it to be a more accurate representation of their activity. So ultimately, I moved forward with just using ODBA as my acceleration metric. So ODBA is a dynamic acceleration where you subtract the smooth 3-axis acceleration from the corresponding unsmooth acceleration, and that removes the gravitational acceleration, Then you add the resulting 3 axes values back together. Luckily, I did not have to do that by hand. I was able to plug in all my 3 axes values into the ADBA function in Tag Tools R package. All right, so I had all of my variables. Now it's time to analyze. So beginning my analysis, I did remove dives that were less than 3 minutes in duration. And that's because for these short, shallow dives, the physiological constraint of oxygen conservation just wasn't as severe. So I didn't believe that they kind of were pertaining to my question. So I removed those. I then used linear mixed effects models, specifically the LME function in the NLME R package. In each of my models, CLID was a random effect to account for the lack of independence of multiple dives stemming from one individual seal. I also added a correlation structure to each of my models, specifically core AR1, that accounted for the autocorrelation in my time series data. So I had 5 dive level models where I tested the mean dive ADBA versus either the maximum dive depth and duration as my independent variables. And those I tested against the following dependent variables: the mean dive heart rate, the mean bottom phase heart rate, the mean pre-dive heart rate, the mean post-dive heart rate, and the post-dive surface interval. So those were my 5 dive level models. For my fine scale analysis, I first divided each dive into its 3 dive phases: descent, bottom, and ascent. I further divided those phases into 20-second intervals. So each 20-second interval's mean ADBA and maximum depth were the deep or independent variables that I tested against the following dependent variables: the mean interval descent instantaneous heart rate, the mean interval bottom instantaneous heart rate, and the mean interval ascent instantaneous heart rate. So those are my 3 fine scale models. All of the models I've mentioned had the residuals tested for homoscedasticity and normality, and I chose the best fit model based on AIC scores and effect sizes. All right, that was a lot. Let's get into what I found. Some general dive summaries. I had 5 females and 1 male in my analyses. All females are 1 year old. The male was somewhere between 1 and 2. I analyzed 924 dives. The mean dive depth was 159.5 meters, and the deepest dive I analyzed was performed by a seal named Bikel at 692.5 meters. The mean dive duration was 17 minutes and 6 seconds, and the longest dive I analyzed was 37 minutes and 6 seconds, performed by the seal named Frixel. And for the surface interval recovery time, the mean for that was 2 minutes. And I actually only had 7.5% of dives that resulted in an interval time greater than 3 minutes in duration. A quick activity summary here. Activity increased as the phases progressed, where the descent phase had the least amount of activity, which makes a lot of sense as seals or northern elephant seals passively descend into the water column. So once they become negatively buoyant, they do this falling leaf pattern as they descend into the water column, really maximizing their oxygen conservation and not having to use a lot of energy or activity to get down to depth. The bottom phase had a bit more activity going on, and then the ascent phase had the highest level of activities, which makes sense because seals, once they want to ascend to the surface, have to fight that negative buoyancy and stroke a lot more frequently. Quick heart rate summary here. I have these first 5 bar graphs are the different parts of a dive, starting at the pre-dive, moving into the 3 phases, and then the post-dive here at the end. You can see with both the pre-dive and the post-dive, they are just over 100 beats per minute. For the different phases, their heart rates actually did not vary too drastically from another. They're all very similar through these 3 different phases, just over 40 beats per minute. And similarly, for the mean dive heart rate and mean instantaneous heart rate, those were both just over 40 beats per minute as well. And I just wanted to point out here that the lowest heart rate I found was a minimum instantaneous heart rate of 4.1 beats per minute. This figure here is what my process data ends up looking like. And I have on the x-axis here the duration of the dive and time. On the lowest graph in red is the dive heart rate. In the middle here in blue is activity or ADVA. And at the top is the depth of the dive in black. And you have longer, deeper dives here on the left-hand side and shorter, shallower dives here on the right-hand side. And I just want to point out here that in these deeper dives, activity is very low on the descent phase, again, because they're doing that falling leaf passive descent into the water column. You can see some peaks and bursts of activity and changes in heart rate going on during the bottom phase of the dive. But then as the seal begins to ascend to the surface, the activity becomes a bit more consistently high until they reach the surface, where they have that anticipatory tachycardia right before they surface, where they increase that heart rate. On these shallower dives, you have a bit more consistently high activity throughout, and that heart rate is just barely higher than those deeper dives as well. And ultimately, what I wanted to show in this figure is the importance of doing fine scale analyses, because when you're measuring the dive on the dive level as a whole, you may be missing these little parts here where there's fluctuations in heart rate and changes in activity and how they may be affecting the dive response on a fine scale. So jumping into my models, my first hypothesis, just to remind you, is that the dive heart rate would be lower on longer, deeper dives, but it would be higher with more activity during the dive. So this first graph is just dive depth versus heart rate, where depth is here on the x-axis and dive heart rate is on the y. And you can see there's a very strong relationship between the two, where an increase in dive depth results in a decrease in the dive heart rate. And maximum dive depth alone explained 43% of the variation in the mean dive heart rate. When we add activity into the picture, we have activity here on the x-axis and dive heart rate here on the y. The color is coordinated by the maximum dive depth, where gray is the shallowest dive, and then moving to orange, blue, green, and then yellow are the deeper dives. So you can see in these intermediate dive depths, there's really not much of a relationship here between dive heart rate and activity. On these shallower dives, there is a positive relationship with an increase in dive activity, increasing the dive heart rate. However, there may be some influence from the surface going on in these shallower dives. And then in these deeper dives in the yellow here, again, there's a positive relationship where an increase in dive heart rate or in dive activity results in a increase in dive heart rate. So it's hard to say, maybe there's some exercise modulation going on there, but ultimately the sample size is pretty low in those deeper dives. So it's hard to say exactly what's going on there. And actually, dive activity alone only explained 5% of the variation in the dive heart rate, not to mention that an increase of 1 meter per second in ADVA, which was some of the largest increases I saw, where the range in whole was only 0.02 to 2.3. So an increase of 1 meter per second is pretty substantial. That resulted in a change of 3 beats per minute in the dive heart rate. So not much happening there. So ultimately, because of that low effect size and because activity doesn't seem to explain much of the variation in the heart rate, I don't believe this is a really important relationship here, and that dive depth actually has much more of an influence in the dive heart rate. And just to drive that home, I looked at that bottom phase dive heart rate versus dive depth and activity to get rid of that surface interference with the dives. And I found that there's no relationship between the bottom phase dive heart rate and dive activity. And dive depth had a very strong influence on the bottom phase dive heart rate, where again, you can see with increasing dive depth, that bottom phase dive heart rate is decreasing. And the dive depth explained 39% of the variation in the bottom phase dive heart rate. But what if you look at the dives at a fine scale? So my hypothesis, just to remind you, is that on the heart rate would be lower with longer, deeper dives, but it would be higher with more activity. So starting with the descent intervals, I found that an increase in dive depth did result in a decrease in instantaneous heart rate. However, activity wasn't as clear of a picture. An increase in activity in depths less than 100 meters or greater than 300 meters resulted in an increase in instantaneous heart rate. And I think that could be attested to for those shallower intervals, less than 100 meters. They're probably still getting some swimming against their buoyancy factoring into that equation there. But during those deeper intervals, greater than 300 meters, there could be some bursts of activity resulting in exercise modulation. But again, the sample size in those deeper depths are much lower. You can see here, there's thousands of samples in the other intervals and not quite as many with the deeper intervals. So it's hard to say exactly what's going on with that relationship. For the intermediate levels, 100 or intervals 100 to 300 meters, an increase in activity actually had a decrease in instantaneous heart rate. And what I attribute that to is that passive descent into the water column. When they're negatively buoyant in the water column and doing that falling leaf motion, there may be some changes in their 3-axis acceleration that the tag is picking up, but they're really not expending much energy during that time. And they're mostly just concerned about conserving their oxygen stores. So I think there might be some overestimation there going on with the activity. And ultimately, their heart rate is decreasing with that increased dive depth. When we moved into the bottom intervals, it became a lot clearer. So an increase in dive depth resulted in a decrease in instantaneous heart rate again. But with activity, an increase in interval activity resulted in an increase in instantaneous heart rate. And it's a lot clearer in this picture here. You can see all of these positive slopes in this graph. And even this really steep slope here with the depths greater than 500 meters, though I do attribute that to only having 96 intervals in that sample. So that's probably why that is so steep. But ultimately, there's a clear relationship, clear exercise modulation going on in this bottom phase heart rate. And the bottom phase or bottom interval activity and depth explained 59% of the variation, bottom interval instantaneous heart rate. So my last fine scale model was for the ascent intervals. An increase in depth, again, had a decrease in instantaneous heart rate. You're going to be tired of me saying that. An increase in activity, again, had an increase in instantaneous heart rate. And that made a lot of sense with the ascent intervals because, again, they're actively stroking back to the surface on the ascent. And as they're reaching the surface, their heart rate is increasing with that anticipatory tachycardia. In the last 20 seconds of their dive, they increased their heart rate by 26%. Moving on to my third hypothesis, that the post-dive heart rate would be higher after longer, deeper, and more active dives. I found that to actually not be true. The post-dive heart rate was not influenced by dive behavior. And let me explain. I did have a significant relationship where increased activity had an increase in post-dive heart rate. However, there was no relationship between dive duration and post-dive heart rate. I still don't think the post-dive heart rate is influenced by this activity because there is a low R squared where dive activity and dive duration only explain 3.6% of the variation in the post-dive heart rate. And again, a doubling of activity from 1 meter per second to 2 meters per second only resulted in an increase in post-dive heart rate of 13 beats per minute. Though that might sound high, that was only a 12% increase. So ultimately, I don't think that's very biologically relevant. And I don't think the post-dive heart rate was influenced by dive behavior. My fourth hypothesis, that the surface recovery time would not change based on dive behavior. I found that it is to an extent where an increase in dive duration did result in a post-dive surface interval that was longer. There was no relationship with activity and post-dive surface interval, but there was a significant interaction between duration and ADVA. Dives that were greater than 15 minutes resulted in a 51% increase in post-dive interval duration, but that was only a 45-second increase to a recovery time of 2 minutes and 13 seconds. So these seals are still remaining typically under that 3-minute mark. So that's why I say only to an extent. And I think that's ultimately because they have such a strong dive response and their dive behavior throughout the dive may reduce their need to recover as they conserve their energy during or oxygen stores during that descent phase. They have that fine scale exercise modulation. There might be things they're doing to reduce their need to recover longer. And lastly, my last model, that the pre-dive heart rate would be higher before longer, deeper dives. I did not find that to be true. I did not find that the seals anticipate their dives. So although, again, I had a statistically significant model where an increase in dive duration resulted in a decrease in pre-dive heart rate, and activity less in dives less than 50 minutes or greater than 25 minutes, an increase in activity resulted in a decrease in pre-dive heart rate. And intermediate dives, 15 to 25 minutes, an increase in activity resulted in an increase in pre-dive heart rate. You can see in this graph here on the right-hand side, there's really no clear relationship going on with any of these intervals. The colors are all scattered about, the lines are all scattered as well. So although it was a significant finding, again, that R squared is so low, ADVA and duration only explained 4.5% of the variation in pre-dive heart rate, and a decrease of 1 beat per minute in the pre-dive heart rate corresponded with an increase of 10 minutes in dive duration. So a 10-minute increase in dive duration only had a decrease of 1 beat per minute in the pre-dive heart rate. And similarly, with a 1 meter per second increase in activity, there was a 11 beat per minute decrease in the pre-dive heart rate. So just not a lot of change going on. And I test that to maybe they're diving within a range of a duration that doesn't require them to need to anticipate it. Or maybe, again, the spine scale, exercise modulation, and other dive behavior are reducing their need to anticipate their dives. All right, so that was a lot of information. Let's discuss it. Going back to that first hypothesis, I found the dive response is stronger in longer dives and deeper dives. Longer, deeper dives resulted in a lower heart rate, and activity had little effect on the dive response at the dive level. However, when I broke down the dive into fine scale analysis, I found that the dive response was more influenced by depth and duration, but there was a small degree of exercise modulation. And the descent intervals, there was more of oxygen conservation going on, but during those bottom and ascent phase intervals, we did see that exercise modulation. And during this time, they are perfusing their muscle with oxygenated blood, reduce that lactic acid buildup, and ultimately maximize their aerobic metabolism. So these findings with my first and second hypotheses are not consistent throughout species, where we mentioned earlier with Waddell seals, they found that an increase of stroke frequency corresponded with an increase in heart rate. And Waddell seals are another deep diving pinniped, and they spend about 5 minutes recovering between dives. They usually go on 12-hour bouts of diving before returning to the ice to recover, which is different than elephant seals that usually spend days out at sea diving. And then with harbor porpoises, for example, in Danish waters, they're performing very short, continuous dives. So very different than deep diving pinnipeds, but similar with northern elephant seals in that they're continuously diving. And they were found to have not quite as a strong dive response as these deep diving pinnipeds. It's not nearly as pronounced, but they looked at their activity versus heart rate in McDonald et al. In 2021 and found that they didn't find any exercise modulation. So you can see with the interval heart rate here on the left-hand side and their activity here on the x-axis, increasing dive duration as you're going from left and right here. These colors correspond to different individuals, and there's really no steep slopes going on with increased activity having any effect on increased heart rate. So the heart rate's staying pretty consistent, although the activity is changing. All of this to say that northern elephant seals kind of fit in here as they're a deep diving pinniped and they're diving continuously, but they have that really strong dive response, and they didn't have exercise modulation on a dive level, but they did have it on a fine scale. So clearly the regulation of the dive response and the severity of the response is different from species to species. And what we're learning is it's hard to make these broad generalizations, and it's really important to study species-specific data. Going back to my third and fourth hypothesis on recovery, I found that there is no change in the post-dive heart rate for these seals, but they did recover longer after longer dives, but only 45 seconds longer. So it still usually remained below that 3-minute mark. Only 7.5% of the dives resulted in a recovery time that was greater than 3 minutes. And I think this is likely due to the active regulation of their dive response during the dive, that conservation, again, on descent phase, their fine scale exercise modulation, and that anticipatory tachycardia right before they ascend. And all of these things just maximize their conservation of their oxygen and maximize their foraging time. And lastly, for the pre-dive heart rate, I did see some degree of cognitive control, though it wasn't with the pre-dive heart rate. They were not anticipating their longer, more active dives, but they had that anticipatory tachycardia right before they surfaced, which reduces their recovery time at the surface as well, because right when they hit the surface, they're ready to go with the gas exchange. And ultimately, that reduces their vulnerability to predators, as they don't have to be up at the surface for a long period of time. So in conclusion, northern elephant seals have a plastic dive response. It's predominantly dictated by depth and duration, though we did see exercise modulation on a fine scale and anticipatory tachycardia again before surfacing. And this ultimately, this plasticity will likely grant them capacity to adjust to climate change and anthropogenic stress, such as prey distribution changes. And though we can apply what we've learned with elephant seals to other deep diving species, there should be species-specific data collected, and it's important to analyze this data on a fine scale. As you saw today with this study, if I hadn't analyzed my data on a fine scale, I would not have seen that exercise modulation that was so clear in those interval phases. And just to touch on some future work, once technology advances a bit, it would be exciting to measure these data on adult northern elephant seals. Although juveniles are similar in their dive behavior and response as adults, adults just have a larger oxygen storage capacity. So it would be interesting to know, with their increased dive depths, does that lower the effective activity? Or is there greater exercise modulation going on at a fine scale or even a dive level because they're diving for longer and deeper? And additionally, getting a diversity of dive types. All of the dives I analyzed were transit dives, but getting things such as foraging dives and different foraging grounds would be very interesting. All right, so with that, I'm just gonna get through some acknowledgements really quick. There's a lot of people to thank, but I'll try to do this quickly. First and foremost, Gita, my advisor, I'm so grateful that you accepted me into your lab and gave me these incredible experiences with different people and different species. I've gained so much knowledge through your leadership and mentorship, and I'm definitely a better scientist because of you. So thank you so much. And hopefully we get to work together someday in the future, hopefully with less tag data issues and better, prettier data and maybe less falling, but you tend to fall a lot. So I don't know if that's reasonable. And then Cass and Jim, my other committee members, thank you so much for all your help and support through this journey. You're so inspiring and motivating. Cass, it was so great to learn from you in the field and see your fixation on detail. It was just really great working with you. And thank you so much for always responding to my emails and meeting up with me here in San Diego and giving me so much great advice. And Jim, thank you for continuing to mentor me, although you're gearing up for retirement. Sorry it took so long to get through this, but thank you so much. Your perspective, your input, they were so important in telling this story. So thank you all so much. There were 7 PIs on this project: Gita, Cass, Allison, Paul, Marcus, Holger, and Dan. You all bring such interesting crafts to this project, and it was really incredible to work alongside you and learn from you. So thank you so much for all of your insight. Lauren and Daphne, the other students on this project, man, I couldn't have gotten through this without you, your guidance, your help, your support. Lauren, thanks for always answering my millions of questions that I send you. And then we had such a large team with this field project. And I just want to thank you all so much for your help. We could not have done this study without all of your hard work. And hopefully I'm not missing anyone here in this list. I had a lot of statistical help, especially from Mason, Parker, and Katie. Thank you so much for meeting with me when I didn't know what to do with my noisy, crazy data. I really appreciate your help. To my RAU students, Alex and Rebecca, for helping me analyze or process my heart rate data. And then to the MLML staff, past and present, thank you so much for helping me along the way through MLML, specifically Tara for always supporting us students and giving us a voice. And for Rhett and Michael for giving me that job at the IT help desk. It was a great way to integrate into the community. Some quick acknowledgements for my funding sources. I couldn't have done this work without them. National Science Foundation, Moss Landing Marine Labs, WAVE Award and Simpkins Award, the Coast Graduate Student Award, American Cetacean Society, Monterey Bay Chapter, and then the Myers Trust Grant. Thank you so much for your support. And just to the incredible Vertebrate Ecology Lab, the best lab at Moss Landing, if I do say so myself, and your continued friendship and support along this journey. These friendships will last a lifetime, and I really hope we get to work together someday. Just to shout out that support, I have Nina and Isaac here with me in my apartment. They literally drove 7.5 hours to come be with me on my defense day. So didn't think I was getting emotional about that, but love them. And the other support at Moss Landing, the community is great. All of the friends I've made along the way, and the funny laughs we had. Thank you so much. And to Matt Elliott, one of the best things to come out of my journey through Moss Landing, in addition to the degree, but he's pretty great too. His support literally got me to this finish line, and thank you so much for being my rock, and I love you. And then to my friends outside of Moss Landing, thank you for asking me about how long it was going to take me to finish this degree and when I was going to be done, and just realizing that we get there when we would get there. And now we're here. We did it. Thank you so much for your love and support. And to my incredible family, I love you so much. And so with that, I'll take any questions.
Great job, Shawn. And I am happy to moderate if there's any questions, but that was wonderful to see your talk. I see you have some clapping hands in the participant tabs. I haven't seen a hand yet. Okay, lots of clapping. I guess I can ask a quick question. You actually answered them. Like, it was kind of funny. Obviously, we did a practice talk, and a lot of my things I was planning on asking you dealt with. But I guess one of the questions you mentioned is like, it'd be nice to see what they do in different types of dives. Have you thought about that? How might it be different in a transient dive versus a foraging dive?
Yeah. So with those foraging dives, they are just spending a lot more time down at depth, so that they would have an increased depth and duration of their dive because they're actively searching for prey. During these transit dives that I looked at in my analysis, they're really just trying to get from point A to point B. They may find an opportunistic fish to munch on or something, but they're not really looking for prey per se. So during these foraging dives, they probably would just have deeper dives and longer dives. So just be interesting to see how that played into that exercise modulation and activity.
All right, looks like we have a question from Jim. I think someone will unmute you. I can guess what this one might be.
I don't know. Maybe you can't. Good job, Shawn. Nice job with the thesis and with the presentation. I've got two questions. One of them is for the 4 species where they actually measured ADL, was there a calculated ADL also? I was just curious how similar maybe the calculated was to the actual measured ADL.
Ooh, I think they used the measured ADL to get their equations for the calculated, but I actually don't recall from that paper exactly if they had the comparison there.
Okay. Yeah, I was just curious because I mean, obviously, lots of people are doing calculated ADLs now because it's so hard to measure it directly. And I'm was just curious to see how similar those two measurements would be. So the second question though is a theoretical one, and that's and I don't know that you won't know the answer exactly, but at least you'll give us some assessment of what you think. Do you think marine mammals and maybe I'll just ask it about elephant seals. Do you think elephant seals have maxed out on their dive response? In other words, do you think there's any more evolutionary breadth to the dive response?
Do I think that they're reaching that max like normally, or that they evolve?
Have they reached their evolutionary maximum on what they can do? So for instance, in another 10 million years, if you were around in 10 million years from now, and you were able to go out and do the same thing, do you think you'd find the same response? Or would the animals have increase their myoglobin content or have a greater blood volume or whatever?
I like this question because there's really no wrong answer. So this is great.
Yeah, exactly.
I believe that they probably could continue to evolve. And I say that because these elephant seals they're not seeming to max themselves out right now. It seems like they have a lot more capacity than they're putting themselves to regularly. So if they got to a point, like I mentioned, with climate change and anthropogenic stress, pushing them to a further limit where they might have to max out that capacity a bit more than they usually do, I could see that resulting in more advanced evolution with their dive response or adaptations that would lead to them having even more capacity than they do now, because I believe that they'll probably respond pretty well to changes that we will see in the ocean. But I do think that could lead to maybe some evolutionary change.
Okay, cool. So sorry, I guess I have another question that's related to that. So given that response, that yes, you think they still have the capacity to evolve a stronger dive response, which component do you think is the one that has would be the one that would most likely work to increase the dive response? So is it vasoconstriction, bradycardia, blood volume, myoglobin or hemoglobin changes? What?
So we actually, when the dive response was first measured in marine mammals, it was during forced submersion studies. Right. So their dive response is not nearly as strict as we saw when we first measured it. So I think it would probably be a more strict bradycardia where the heart rate would maybe get even lower. But I could see also that oxygen capacity, the oxygen storage, possibly evolving as well there. But I would say probably just the dive response would likely be the first thing we would see adaptation wise, because they're not having like as strict of a response as we know that they're capable of. So if they were pushed to that point, they may start having more adaptations to have a even more increased bradycardia, more vasoconstriction going on. But that may lead to them having an increase in their oxygen storage capacity too.
All right, cool. Thank you.
Yeah.
All right, and it looks like Cass has a question for you. Your committee is hitting you.
Committee.
Yeah.
All right, good job. That is such a complicated data set. So good work with that. And I'm going to ask you a question, I think, about that complicated data set. So correct me if I'm wrong, but I think you said that in your interval analysis, the relationship between instantaneous heart rate and bottom phase was the strongest, like the highest marginal R squared. And I wondered if the effect of interval on OBDA was looked at. Like, was the bottom phase, did it have the highest OBDA and maybe, or the largest range? And maybe that plays a role in why we see that heart rate that they're modulating at that time, because that's when they're most active. Does that make sense?
Yeah, I could see that. I mean, they're definitely most active during that ascent phase because they're stroking more frequently. But you mean just because like the variation from being not active, stroking and gliding and not being very active at all to like stroking more frequently? Is that what you mean?
Yeah, I just wondered, it was interesting that that was the phase that had the highest relationship, or R squared.
Yeah, I can't recall off the top of my head if the range was highest with the bottom phase heart rate. I could see if I could go back to that graph, but actually, see here, there's a way to do that. Yeah.
Or maybe behaviorally it's the most unpredictable, because if they're foraging or messing around,
Yeah,
doing something different on the bottom, then
yeah, I think that probably has a lot to do with it because they're so strategic, and the two other phases, like descent phase, is so oriented around conserving their oxygen. And it's even been seen that they sleep during that descent phase. So they're really minimizing their effort in order to conserve their oxygen during the descent phase for the rest of their dive. And then the ascent phase is so concerned about getting back to the surface. So the bottom phase may be where they're doing a lot of active regulation because it is so variable. And again, that's why foraging dives would be so interesting, because there would be a lot more going on at the bottom phase than just in these transit dives. But let's see. So yeah, I mean, the activity was about 0.02 up to, yeah, about 6 meters per second squared. So that is probably the highest range I saw between the 3 phases. So that could be what's going on, is that there's such a variation between not being active at all versus having these short bursts of high activity if they're quickly,
right,
frequently all of a sudden. So that could be why there is that really strong exercise modulation going on in the bottom phase of the dive.
All right, great. Thank you.
All right. Any more questions? Otherwise, we can let Shawn off the hook, and then Cass and Jim and I can always ask her more questions. All right. Well, then we will call it a day